In this video lecture, we will discuss about our new topic, which is an study of microdiversity. First of all, what is biodiversity? Biodiversity is the variety of plants, organisms or animals living in the specific habitat or the place, which is termed as an biodiversity. The word itself indicates the bio means living organisms or living things, and diversity means variety of the specific species living in the habitat. Here we will discuss about the study of the microdiversity. Microdiversity means the study of the diversity of different microorganisms. There are the several methods to study the biodiversity or microdiversity. First method, which is an microscopic method, another method is an cultural method, and the last method which we will study is the phylogenetic method. First of all, we will discuss here about, in this lecture we will discuss about the microscopic method for the assessment of biodiversity. Here we have microscopic method means here we will discuss about, or we will see about the different types of microscopes which is used to assess or which is used to study the microdiversity. First of all, introduction. Here the microscopes are the traditional instruments used for assessing the microbial diversity. Assessing means to study the microbial diversity, and they remain indispensable tools for exploring the morphological, physiological and genetic diversity present in the microbial communities. What are the microscopes? Microscopes are the instrument which is used to study the microbial diversity, and they remain indispensable. Indispensable means that they will not go out of view or will not be dispensed. Tools for exploring, which is what is use of microscope. The microscope is used to explore the morphological, physiological as well as genetical diversity present in the microbial communities. Now, what are the criteria before using the microscopes to study the microbial diversity? There are 5 different types of criteria involved to study the microbial diversity. First criteria, which is an advances in the instrumentation and methodology for assessing the microbial diversity. Advances mean that they need an advancement or enrichment in the instruments and methodology for assessing the microbial diversity, in this means we need the any enhancement or further advancement in the instruments here, which is resembles the microscopes. And the methodology, method means different types of microscopes leads on the or work on the different principle for assessing the microbial diversity. Second criteria, which is the breadth of the microbial morphological diversity in the microbial communities as revealed by the microscopic analysis. The breadth of morphological diversity means there is a large type of, or means it is a large community which of the morphological diversity in the microbial communities. There are different types of large number of diversities or morphological changes in the microbial communities, which is used or which is revealed by the microscopic analysis. Third criteria, which is a limitation of microscopy. What is one simple microscope has its own limitation. Every single microscope or microscopy has its own limitation for further not exploring or magnifying in the specimen for investigating morphology as the fundamental basis of a species classification. Every single microscope has its own limitation. The fourth criteria, which is the suitability of microscopic analysis, on which specimen, which microscope is suitable for studying the specific specimen which we need to assess it or which we need to know it before studying the specimen. Here the specific microscope is used to study the specific specimens or the specific microorganisms for assessing the microbial cell structures, colony architecture or colony characteristics and community organization. Here it is an suitability of microscopic analysis. Suitability means a suitable microscope is only used for the specific specimens which we need to study. The fifth criteria, which is an approaches that are compatible with microscopy for assessing the diversity in various microbiology ecosystem. Here the approaches which is need to study or which is need to assess the microbial diversity in the ecosystem. The ecosystem includes the various aquatic ecosystem, terrestrial ecosystem, aerobic and biological samples. Here the approaches means the assessing the diversity, which types of diversity is need to be assessed or which is been assessed in the microscopes, such as aquatic diversity or aquatic ecosystem, terrestrial, aerobic ecosystem, as well as biological samples are been assessed or been studied using the microscopes. There are different methods for studying the microscopic method. First one is an bright field or basic light microscopy. In basic light microscopy, it involves the compound microscope, dark field microscope, as well as phase contrast microscope. These three microscopes are involved in the basic light microscopy. The another one is an electron microscopy which involves the transmission electron microscope, scanning electron microscope and many other microscopes. Here the third and the last one microscopy which involves the specialized light microscopy which involves the confocal laser microscopy. These three types of microscopy are being used in the study of microscopic method to study the microbial diversity. First of all, we will see about the basic light microscopy. Basic light microscopy which involves the compound microscope, phase contrast microscope as well as dark field microscope. On which principle this light microscopy works? The microscope cannot resolve the details of the object smaller than the wavelength of the electromagnetic radiation. The specific microscope are been designed according to its wavelength. According to the specific wavelength, those microscope can magnify the specimen. Of more than that wavelength, the microscope cannot magnify the specimen or the image. What is an electromagnetic radiation? Radiation means the waves and electromagnetic means the electron and magnetic field. Electricity and magnetic wave is been involved. The waves involved in the electromagnetic radiation are the waves, X-rays, gamma rays, UV rays and infrared rays and visible light which involves in the electromagnetic radiation. The wavelength of the electromagnetic radiation spectrum in which the specimen is been viewed. The average wavelength of white light is an 550 nanometer. More than that nanometer, this microscope cannot magnify the specimen. The white light means the visible light. Therefore, the size limit of the specimen that can be resolved with the microscope, the size of the limit or the limited size would be resolved or can be magnified with this microscopes that depend on the visible segment of the electromagnetic spectrum across the 200 nanometer. What is the spectrum, electromagnetic spectrum which is been designed on the light microscopy, which is an across approximately 200 nanometer. Here where the oil immersion, why we use oil immersion lens? Here the oil immersion lens is used to reduce the wavelength of the light by factor 1.5 to 1.8. It will reduce the wavelength the more the wavelength, it will reduce up to the 1.5 to 1.8 factor. The compound microscope consists of the both objective as well as ocular lens. Objective lens, which is an magnifying lens, and ocular lens, which is also known as an eyepiece, which is used to magnify the image to the eye. Instead of the single magnifying lens, in the simple microscope only single magnifying lens is being used, while in the compound microscope objective lens as well as an ocular lens has been used. Now we will discuss about the phase contrast microscopy. What is an phase contrast microscopy? The phase contrast microscopy is an another modification of the basic light microscopy. Phase contrast means it will contrast the specimen and the light, which will give one final image according to the light source. It will magnify, or it will specific magnify the image, and it will give the phase contrast. Contrast means it will clear the image according to its background. It is another modification of the basic light microscopy, which enhance the visualization of the internal cell components. In the phase contrast microscopy, the internal cell components is used to be assessed, or it is used to study the internal cell components. The variation in the density of the organelles, what is the density here? If the density variation in the density of the organelles influence their interaction with the incident light. Here, if the density of the specimen or the organelles is been influence their interaction with there is the variation between the density of organelles and interaction of incident light. This is all about the phase contrast microscopy. Now we will discuss about the another microscopy which is involved in the light microscopy. First, this is an dark field microscopy. What does dark field microscopy discuss? Here it is used to visualize the transparent cell. This is based on the principle of the dark field microscopy. If the cells are been transparent, then there is no need of any light source, because the light source will not get visualize our transparent cell. Here it works on, here it is similar to the negative staining, in which the dark background will be in dark and the cells will be transparent. It is used to visualize the transparent cells by reducing the background lightning and around those cells. It will reduce the lightning on the background and around those cells, through which we can visualize the transparent cells. The cells are been visualized by the dark field microscopy and appear the bright against the dark background. This is all about the basic light microscopy. This involves the basic light microscopy involves the compound microscope, phase contrast microscope, as well as a dark field microscope. Now we will discuss about the electron microscope. What is electron microscope? Who discovered the electron microscope? The Max Knoll and Ernest Ruska, who built the first practical based on the electron microscope in 1931 at the Technical University of Berlin. Who performed the first electron microscopy, which was the Max Knoll and Ernest Ruska, which performed the first practical on the electron microscopy. Now what we will discuss about the 3 types or 5 types of the electron microscopy which is involved in this microscopy. First one is a transmission electron microscope, scanning electron microscope, scanning tunneling microscope. Here the chemical microscopy is also been involved. The first one, transmission electron microscopy. Here it is been operated in the manner similar to the transmission light. The transmission of light here will play a major role in this type of microscope. This microscope producing the two-dimensional morphological information. This type of microscopy which will generate the two-dimensional structure. Here on the basis of the atomic diameters. The Transmission electron microscope facilitate the observation of the crystallography. Here it will facilitate the observation of crystallography as well as the biochemical details with the few nanometers. Here it will facilitate or it will magnify the observation of crystallographic as well as an biochemical details among the few nanometer areas. The next microscope, which is an scanning electron microscope. Here it will visualize or it will give the three-dimensional image. It will visualize or magnify, and the real final image will be the three-dimensional image produced by this microscope. Here, through which the three-dimensional microscope is been giving the three-dimensional structure or been image. Here with the aid of the electron instead of the light waves. Aid means with the help of electrons, this will produce an three-dimensional magnified image through which through electrons it will give us the three-dimensional magnified image. The other microscope, which involves the scanning tunneling microscope, atomic force microscope and chemical force microscope. In the scanning tunneling microscope, it is an improved version of the development of, it will improve the resolution power in this type of microscope where the atomic white tip is being used. Here the atomic tip is being used to scan this specimen surface from the height of approximately, we used to say that it will approximately according to the 1 nanometer, it will use to scan the specimen. Now in the atomic force microscopy, the scanning here it is a modification of the scanning tunneling microscopy. It is the variation in the scanning tunneling microscopy. In this, the semiconductor tip is been used. Well, in the scanning tunneling microscopy, the scanning tip is been used, scan tip is been used. And in the chemical force microscopy, it is the use of different chemical forces across the specimen surface of the molecular level. Here in the chemical force microscopes, the chemicals is been used across these specimens to give the magnification or to give an specific magnified image. The last microscopy, which is an specialized light microscopy. Here in this microscopy, confocal laser microscopy is been used. Confocal means from the single focus or from the single area the laser is been passed. Confocal means that from the single focal or means single focus has been used extensively to investigate the microbial phylogenetic diversity. The confocal laser microscope dissects artificially stained fluorescent specimens by using the light sources at the different wavelength. Here in this microscope, it will dissect the artificially stained fluorescent specimen. It will dissect the fluorescent specimens by using the light sources of different wavelengths. The stains such as digoxanine, diamidino, 2-phenyl, indol, 2-HCl and 5-cyano-2,3-dithiol tetrazolium chloride dyes are being used to paint or to stain the specimen, which is an fluorescent dye. Through the fluorescent dye, they will dissect the fluorescent specimens by using the light source and the different types of wavelength. Here in this type specialized light microscopy, the fluorescent microscopy is been also involved. The typical confocal laser microscopy is been equipped with the laser source. Here the name itself indicates it will use the laser sources to emit the different wavelengths such as argon and helium. The wavelength of argon will be around 458 nanometer to 488 nanometer, while the helium wavelength will be the 543 nanometer to the 633 nanometer. When focused, this laser wavelength, which will focus on the fluorescent specimens, the laser will provoke, or the meaning of provoke means it will excite the emission of high energy photons that are detected by the visible light. The fluorescent specimens will be focused by the laser. The laser will provoke the emission of the high energy photons that are detected at the visible light. This is all for the microscopic study or the microscopic method used for the assessment of biodiversity.